Thank you, Jens. Great to see you, pal.
Morning,
Alan, David. Thank you so much. Roberto, thank you. Dennis, thank you.
There are names.
Yeah, because I get confused with names. So Amy, if you're in the audience, could you bring my jacket up? I left my questions in my jacket. If I could, maybe starting with you, Dennis, if you could each introduce yourself and maybe any thoughts you have on the work you're doing, just to give them a little bit of an insight and a perspective, that'd be awesome.
Okay, I'll start. My name is Dennis Kirik. I work at Lund University in Sweden. I'm a medical doctor by training, but have been doing over 2 decades work related to neurodegeneration, Parkinson's disease in particular, and development of, let's say, non-small molecule drugs for treatment based on cell and gene-based treatments. So I'm not an technical assay development person. That said, everything we do relies on analytical tools. Their accuracy, availability, and robustness determines whether we actually can be successful or not. So it is in that angle that I've actually spent quite a bit of our time on working with biomarkers. And in the context of that, I'll come back to specifics of
You're going to be presenting later today as well. Is that correct, Dennis?
Well, I've actually made a better deal. So I said I'll give some insights into what we're doing right now, and you'll invite me again for a second meeting where I'll make a presentation.
Awesome. Liesbet?
So hello, my name is Liesbet Peeters. I am a data scientist from Hasselt University. The lead investigator and co-initiator of the Multiple Sclerosis Data Alliance, which is a multi-stakeholder initiative that aims to transform the care of people with MS through better use of health data research. Because we believe that to transform the care of people with MS into a next generation of management that is personalized and at a precision level, an extensive amount of data is required and collaborative research is necessary. And data sharing and collaboration is not easy. And that's why with the MS Data Alliance we aim to overcome the sociological as well as the technical challenges that arise when we want to scale up health data research in a multi-stakeholder setting. So we are now in the process of defining and finalizing the consortium to kick off in 2019. We have more than 30 registries and cohorts that are interested, more than 20 patient societies. We have the European Medicine Agency, some umbrella organizations, and we have the potential to combine or at least collaborate 300,000 patient trajectories if we are successful. So that's the mission.
Where are you located?
So actually today it's a joint initiative between Hasselt University and the European MS patient platform. The idea is that when we kick off, there will be a legal entity, and then it still has to be decided where that will be.
Excellent, excellent. Thank you. Roberto.
I have my my name is Roberto Furlan. I'm an MD, a PhD also, and a clinical neurologist as well. I think I have been in the MS field since 1988, so some time. And we are especially focused on developing new therapies, and with new therapies, experimental therapies, we also need biomarkers. We were actually pioneering some years ago the field of extracellular vesicles in MS as, let's say, theranostics. Do you say that? So both diagnostics and therapeutic tool. And well, I don't really like technology-driven science as a concept. I usually like ideas and scientific question-driven science, but I have to admit that we need extreme technology to advance. So this is why I'm pretty interested.
Thank you. My name is Charlotte Teuniseen. I'm from here, Amsterdam. I'm professor in neurochemistry at Amsterdam VUMC, which is the the merging of the two academic hospitals here in Amsterdam since last June. The focus of the research of our group is to improve patient care by developing good biomarkers, and our focus is on Alzheimer's disease and multiple sclerosis. So what we aim is developing novel biomarkers for diagnosis and prognosis and therapy response for primarily Alzheimer's disease, but also other dementias, and also in the field of multiple sclerosis. So we have a strong focus also on CSF biomarkers, and for which we have a large biobank of over 4,500 patient collections of CSF paired with blood. So that's a gold mine.
You actually have both for all those patients?
We do, yeah. So that's our dementia cohort. So it's enormous. So it's enormous wealth for us to do our research. And besides that, we are a central lab in a couple of trials also for Alzheimer's disease, mainly for inclusion, so doing the Alzheimer biomarkers. Now in CSF, we hope in the future also in blood, and we do endpoint measurements as well.
And what's your current level of belief that you're going to be successful with blood biomarkers versus cerebral spinal fluid? Do you have a
it's not versus. So the blood will be the pre-screening because the CSF biomarkers, they are quite specific and more or less the golden standard, CSF or PET. So we think that the position of the blood biomarkers is in early pre-screening, probably for people that are reluctant to undergo a lumbar puncture. And if there is a positive result, then they can go for further testing.
Excellent.
But my level of belief is 100%. So we will arrive there. I'm convinced.
Very good. Thank you.
The floor to David.
Yes, I agree with you. We will be successful. My name is David Leppert. I'm a neurologist and specialized in translational medicine, mainly in the field of multiple sclerosis for the past 2 decades, both in academia and industry. I was a therapeutic area head in Novartis and initiated a lot of neurofilament light work within the context of phase 2 and phase 3 trials in order to demonstrate that this is a drug response marker among other features of neurofilaments. This focus will remain while I transition now back to the University of Basel, working with Jens Kuhle, with whom I worked in the past 3 or 4 years to expand on the knowledge of neurofilament and bring that from a concept to an applicable biomarker for individual patients in the near future.
Awesome.
Thank you. My name is Henrik Zetterberg. I'm a professor of neurochemistry at University of Gothenburg and University College London. And I work much like you guys also with biomarkers for CNS disorders. We have a team focusing on trying to develop targeted biomarkers for pathologies that occur in the brain in Alzheimer's, but also on some of those other diseases where we currently do not have good biomarkers. There are 2 pathologies we really need to develop sensitive tests for, and that is Lewy body pathology and also TDP43 pathology. And there I think we can make use of ultrasensitive technologies, much like Kevin mentioned, not just to move into blood, but also to make very specific and sensitive CSF-based tests in the first place. And then, of course, if they are working well, then we could try to move them into blood tests.
So thanks, I'm Jens Kuhle. I'm a neurologist. I lead the MS Center in Basel in Switzerland. So we are a center mainly focusing on MS, but also see other patients, also dementias and things like that. But my research has really been driven by the fact that I sit in front of the patient and I don't really know if the drug does its work. And by having quite many drugs at hand in MS. So it's really about, like Kevin also pointed out, cycling these drugs quicker and maybe being able to detect subclinical disease activity. And by being a clinician also, I also see and I'm able to compare with imaging. That is the gold standard really in MS. And it's a luxury situation. I believe that there's so much room for improvement there, really, by being able maybe to cover the whole CNS by doing a blood sampling. And 5 years ago, there was no real good evidence that NFL, for example, could be higher in MS than in control. So I think we've come an awful long way already in the last 5 years.
So Jens, because you are a clinical practitioner as well, I'm curious, where do you think we'll be in 5 years, and what would you point to as being where a lot of our focus needs to be to accelerate the progress that I think intrinsically you believe is there?
Yeah, I just told to my colleagues in the elevator an hour ago that about 1 year ago we wouldn't have dared to offer NFL measurements in individual patients at the moment, but now, just a few weeks ago, we've started a program where we will now offer these measurements for individual patients, comparing it with healthy controls. So I think it's fascinating that there is really an option that these measurements could come to clinical practice in 5 years. I think it's not excluded, which is unbelievable after decades of no progress, especially in diseases like MS, with regards to blood biomarkers. So I do believe this is really a fascinating time, and there's a big realistic chance. Neurologists usually are quite pessimistic, you know. So this is a chance.
So, you know, I would ask the whole group, like, when you look at where your current science stands, what is it that you believe we should be focused on to try to accelerate the next level of practice utilizing the technologies that you're working on? Are there ways and things that you feel are getting in the way as you look at the current state of your work that teaches you what you think is in the way so that all of us can benefit from your views on that? So any of you could start. Dennis, did you?
Okay, we may have different perspectives to reflect on this broad question. If I respond to it from the area and the spectrum I cover and work on, one of the things we would really like to make a difference is when we try to advance a novel therapeutic approach towards, let's say, first-in-man trials, where we want to see, does this have a chance of making a difference? We actually face a number of hurdles in doing that translation. This goes from the validity, reliability, and translatability of the data in animals to results you would obtain in the clinic. It goes from tools and techniques we can use to assess the outcomes, whether what we look at in an animal model is the same thing we can look at in a participant to a clinical trial. It also goes in the following way. So what you actually test in the clinic, what you then market in the future, is what you can make money out of, right? So the constellation of a clinical trial and a drug that is marketed later on may be different than what a scientist may think in the lab is the ideal solution. Either it's not translatable, it's not practical to make a drug out of that, or it doesn't actually compile to a product that can be put forward. So we actually have, we can say, lost in translation in many dimensions. And so the fewer the gaps are, the lower the impact of each one of them, the better the chance for us to actually have a successful new drug that actually makes a difference. And that difference would be, as you described, can you actually interfere with the disease at an earlier stage so the patients don't have to become severely ill, bedridden, requiring nursing home care, keeping them at home, at work, with their families is a significant improvement. But this is not easy to validate in a preclinical setting.
And do you have tight relationships with pharma, biotech that you feel benefits you in advancing that translation?
Yes to both. Of course, I work with non-traditional drugs. Industry has, in the past at least, stayed away from them. simply because they didn't know what the marketing model would have looked like. Now that situation has changed. So over the last 2-3 years, there is a rapidly increasing interest, both internally in their own organizations and their relationships to external parties, including academic researchers, spin-off companies, small biotech firms that are rapidly growing in that space. So what we see today is very different than what it was 5 years ago. So tomorrow, what we will see could be significantly better than what we actually have experienced today. So this is a rapidly changing landscape.
It does be interesting.
Towards the better.
Towards the better. Be interesting as each of you comment on this. Like, how are your relationships with the pharma biotechs, and are they appropriately funding your opportunities that you see, and or are you appropriately linking with them? And do you have sufficient kind of relationships to kind of make sure your work is seeing the light of day? In any order, so please.
So maybe first on the first question, what are the challenges? So coming it from a data science perspective, I believe that many of the challenges that arise today are not really technical. Because in a world where Netflix can predict which movies I will like, which is quite difficult because it ranges from Desperate Housewives all the way to Vikings and Spartacus. So I am quite broad flexibility in that field. So technically it's probably possible to do like a personalized prediction of effectiveness. Why is it not possible as Netflix does it? It's because the data is very awful. It's somewhere most of the time in a basement, in a clinic, in a paper document. So that's why we are trying to focus today on making data fair, which is an abbreviation of 4 words: findable, accessible, interoperable and reusable. And especially in the MS Data Alliance, we are not focusing today on entering the biomarkers field because we are not there yet when it comes to health data. It's really just about standard of care. Most people, if they go to the doctor, most visits consist of just a doctor asking, how do you feel today? So we have to improve standard of care, that a disability score is measured, that the date of diagnosis is recorded, the date of onset is recorded, an annual MRI is measured, blood is being taken, then biomarkers are being measured. That's the first step, and then clinics have to open up that data by making it accessible, interoperable and reusable, and having people sign an informed consent that makes it possible to reuse the data that is just collecting in a routine way in the clinic. So that's especially the challenges that we want to accomplish. And then if we go back to the interactions with the sponsors, we can feel that those challenges are not really sexy to sell, because everybody knows that it has to be done. It makes so much sense that it has to be done, but everybody is looking at the other partners to do it. Pharma is telling us, why can't the European Commission pay it? And they're probably right. And the European Commission is telling us, why isn't pharma paying? Because they have to have the clinical trials. So we wanted to do it in a multi-stakeholder setting because we believe the timing is there. And we have to collaborate it and do it in a multi-stakeholder setting. So that's why we stayed quite broad, but it has made us very challenging to find funding to do the kickoff year. Especially because it's constantly the idea of the chicken and the egg. You need money to start, but you need something to have it funded. So today we feel quite comfortable that we will have enough funding to kick off. And then hopefully once we have something very concrete, we will have more opportunities to get funding. Exactly.
Perfect.
I'd like to comment on your question whether how the interaction is between academia and the pharma or other industries. And I completely agree with you that it's very crucial that we communicate very well in the early stages of development. So, for example, if we develop a biomarker, we need a diagnostic company for further marketing, or to bring to the market even. So all the necessary steps that need to be done before a novel diagnostic marker can enter the market. And continuous interaction with pharma industry is also very relevant to define which biomarkers do they need and which biomarkers do we have, and what are the targets and what are the pathological mechanisms that we wish to have biomarkers for, and that are relevant also through their translation as surrogate markers for clinical symptoms that we think are very relevant for the specific disease. So be it Alzheimer's or MS or any other disease like cancer. And I also completely agree that and I would like to make a plea also that we interact more strongly than we did before in making the translation from the animal model results to the disease situation in humans. Especially nowadays that we have very sensitive technologies, there are more opportunities to look into the body fluids. Well, my main interest is CSF, of course, and if we have only a minute volume that we can obtain from animals in those disease models, it's fantastic that we can now analyze those, the proteins that we're interested in very small volumes due to sensitive technologies. It could be quanterics, but it could also be other technologies that nowadays being developed. So yes, I agree, we need to have a good interaction, especially now because we are moving fast forward in the development of biomarkers in relation to diagnostic purposes, but also in trials. So there must be a good interaction between pharma and academia.
Something you said there, Charlotte, that I think we've been hearing, it came up at dinner last night, is the sample preservation, the ability to get an answer out of a very small sample. Sensitivity can play a major role by just diluting up and still having sufficient, like when you get another 100x, then it might then give you opportunities for that smaller sample to kind of get info. Please, Henrik.
I think this is one reason also for it's such a great field to work in, the biomarker field, because it's so collaborative, and we can send around samples, we can compare methods, we can develop different methods for the same biomarker and see what it looks like. But one thing I think we need to think of, all of us now, is to also make our studies so that we can share samples. And because now with GDPR and all, there are many regulations coming up, so that it's quite hard sometimes to get out data, to send around samples, especially if that has not been part of the informed consent that the patient or research subject has signed. So I think we need, as a field, to be even more proactive towards allowing the studies to be global and collaborative, and allow for sample sharing much more, and that the people who enroll, who get into trials, that they are also getting the opportunity to allow for that, and perhaps even demand it. Because now I have learned that some companies have to destroy their samples because they are not allowed to keep them. They can't share samples, even if the people running the trials are super interested in a new biomarker, for example. There might be legal hurdles making it impossible to do. So that is one thing I think we should work on, trying to prepare all the studies we are initiating for collaborative projects, and that I think is an important part, aside from all the things we need to do in regards to developing reference materials and reference methods and standardization efforts to also increase the comparability of studies.
Yeah, that's the one that keeps hitting me. Is that I hear your name used a lot in Lilly, and when I go to the pharmas, your guys' names get used a lot there. I think they're relying on your insights and your perspectives. But I do wonder about this cross-validation, so that like all the pharmas are having this similar kind of ability to scale. And even in the interactions with the FDA, it seems to me like I'm finding a lot of individual companies working with the FDA trying to differentiate their own position, but they're not necessarily looking across a collaborative effort where it might advance the field quicker if we could get more of a collaborative validation versus each company trying to get there. So any influence you can throw into the boardrooms, Henrik, I would think you should.
I think this is a very important point. It has two aspects. One is the technical Henrik started with. Credibility can be created if a test is valid across the planet. And either we come to a standardization of a specific test, like glucose, there's no question whether in Madrid or in New York it's the same value. For the innovative biomarkers, that is much more difficult. At least we need to create exchange rates between different test systems in order to create credibility vis-à-vis the regulators, vis-à-vis the society. That's a technical aspect. And the conceptual aspect is, as physician scientists, we are satisfied to correlate a biomarker with our easy readouts, for example, brain atrophy in MRI. That's a valid result. But we need to focus on what is patient-focused.
Sure about this? That this is a valid result, brain atrophy in MRI?
I say...
Can I challenge that one?
Yeah, you can challenge it, but I just say what is the current currency we deal with, or what we are limping with. We need to focus on readouts that are undisputable, namely that are focused on the well-being of patients. And if we can link our biomarker to that, for example, cognitive decline, psychological well-being, etc., then we create credibility versus regulators, because that's undisputable.
So, Jens, before you comment, I think you started down a path that I can't wait to hear your views on, and that is that I sense that there's gold standards today that may not be gold standards, but the FDA and others consider them to be gold standards, and I worry that we might have such an uphill battle because we have to somehow compare to gold standards that we might miss the whole field and the whole opportunity area. And something that two of the investors said to us in London, they said, we think the imaging companies are going to strangle you guys. They're so wealthy, they got so much influence, they're going to probably strangle you guys before biomarkers can really ever make it as a gold standard. And so I'm curious how we break through some of that in your mind.
I mean, I don't think we need to be too scared about these comparisons. When we did it in the MS field, I have to say NFL specifically made a very good comparison there. I don't think there is too much to worry about. I mean, what is interesting is that there's tons of data more or less laying around, and we don't have the people to analyze it specifically, right? And to harvest it and to illustrate it. Really about, and this is also when we talk about the FDA, I think one of the hurdles will just be to compile all the data in a systematic manner and getting people to share the data. I mean, I fully agree that these sample data sets collected with a lot of money over many years with great standardized technologies are extremely valuable in the companies. And it's interesting to see how the companies all have their own way of behaving and of being interested in things or not being interested, or doing things very secretly or doing things openly. And I mean, that's a big problem, of course. And I hope it's not completely naive to hope that this could somehow be solved. Because it's incredible treasures that are collected and that are laying around that academia cannot do in such a way. So it's extremely fruitful if such collaborations can take place. But resources is really a slowing down factor also here. I mean, it's crazy how important it is to have people to analyze the data. Yeah.
Before Dennis or Roberto, before you comment, Steve Jobs stood up, I think, 15 years ago and said that we need to collaborate. And he wired in Bill Gates into his meeting, and all the Apple employees started booing. And Steve Jobs says that we will never advance digital technologies unless we collaborate with Microsoft. And everyone was booing, and he said the pace of disruption will be stymied if we don't find ways to cross over. And I watch the pharmas, and I sometimes worry that they're so profitable that they do have sufficient willpower to kind of differentiate themselves, and that might slow down the whole biomarker movement if they do that versus collaborate. But anyway, it just hits me that industry has gone so fast in disruption, and we're very slow, and I worry that we're not going to work together. Roberto?
Can I start from there?
Please.
Because I think, to be a little bit provocative, that in fact, here we are booing at MRI, which I think is wrong. You were so happy yesterday at the dinner because there will be a debate, NFL versus MRI at Controversies in Neurology by David. And I think this is culturally one of the hurdles we have to overcome, because the future is not going to be one biomarker. It's going to be integration between all the available biomarker. Her example is perfect. Spotify that knows me better than my mother in terms of music tastes, because it's able to integrate so many different inputs. And I think to make precision, personalized medicine, this is exactly the need we have. And actually, we are not so collaborative in the field. Actually, there is contraposition with the people from the MRI and the imaging.
No, but there's no doubt we need to compare with the current gold standard, which is imaging and clinics. No doubt. Of course.
And there is not only an economical profitability in imaging, there is also an academic profitability in imaging, because with 20 minutes of a sequence, you get over, I think, 100 parameters for each voxel. So you can make perfect salami science. With a few patients, you can publish 20 papers. You can't do that with a body fluid biomarker. So we need to overcome all these hurdles.
I think, Roberto, you're bringing up an incredible point, and that is there should be someone on this panel from the MRI world, really, right? I mean, because I think that discussion that you're pointing out is imperatively important, and I think that the onus is on us to pull them in, because I don't think they're going to want to naturally be here. So I think we got to be nice to them and invite them in.
I think they have to learn that probably body fluid biomarkers are going to make MRI much more efficient.
Yep.
And the other way around.
Interesting. Very good.
I think that's a very important point. And when we discussed about replacement of MRI, we as biomarker field should not go into a row or a confrontation, but show what can we contribute to qualitatively improve, you think, and reduce your quantity of routine things that maybe you are not so much interested in, but you want to spend your time on academic questions. How can we contribute to that? And then that resolves somewhat what many perceive as a natural confrontation.
It's also interesting, like I remember General Electric was worried about Alzheimer's, and they had the PET scan that's very expensive, and you were mentioning, Charlotte, the possibility of maybe using blood biomarkers as an initial screen. Maybe you could move people into something that's a lot more expensive by having an initial screen, and it creates like a demand profile for that PET scan that today doesn't exist. And so by working together, you might be able to create that 1-2 punch type of opportunity. Dennis?
I was going to say exactly the same. So if I start from this issue about gold standards, I want to give an example from the Parkinson's disease field, where MPTP, intoxicated non-human primates, will be seen as a gold standard animal model. Yet, if you think about the Parkinson's disease in the clinic, there are probably a lot that they share. It's correct that if you intoxicate a human being with the same toxin, they will kill their dopamine neurons and they will become Parkinsonian in appearance. But that doesn't mean that toxin injection in an animal or a human being reproduces Parkinson's disease in the general population. That distinction is rarely made. And if you actually make the statement in an audience of PD researchers, half of it will get up and start screaming at you and leave the room. So there is actually, it's true that they will protest. But the essence is that the validity of the models as gold standards for the clinical case isn't all that great, especially when you think about disease modification. So then moving on to the issue with imaging versus wet biomarkers, the situation with alpha-synuclein as a potential biomarker in PD has a unique advantage in the sense that the MRI is not all that useful to begin with. synuclein-specific PET tracers are struggling. I mean, we and others have worked on it over 5 years. There isn't even a good candidate to take into clinical testing. There will be one beginning next year. I bet you it's not going to be a good one to begin with, and we'll see several iterations of that will have to come in and test themselves, right? The wet biomarker space with synuclein and modifications of synuclein, the post-translationally modified species, could actually lead the way, could prove themselves to be useful, and can then become the reference for imaging markers to validate themselves. So you could actually be ahead of them and then say, if you're going to develop a PET tracer for synuclein, you ought to use these wet biomarkers and either stratify your patients or know what kind of species exist in those cases, so you could actually say, when I have a signal, I have good reason to believe that that comes from what I intend to image.
So Dennis, I find that intriguing because the synuclein example, I look at like these countries that didn't have landlines, and then all of a sudden they got the iPhone and they kind of leapfrogged into a whole new generation. And I look at concussions right now. We've got 4 or 5 companies wanting they've got concussion drugs, but they don't know what endpoint to use with the FDA. They don't have any kind of imaging endpoint, so they're trying to innovate endpoints right now, and maybe you're right, maybe there's lower resistance to go where there isn't imaging, get some established protocols, and then back into imaging. I'd like to open it up to the group. Any questions that we might have, from the group before? I think we still got like 10, 15 minutes. So who has a question out there? Please. Thomas
Hughes, NMI. Jens, you mentioned that we have a lot of patient samples and data material available which is not carefully analyzed. My question to the audience would be, who has ever participated in a clinical trial effort? And if so, have you run all your clinical biomarker efforts according to those standards? My impression is, as mentioned already by Kevin, that we have 19,000 biomarker discovery publications, and a lot of them are just discovery because it's academic discovery. Could you comment on this, Jens?
Well, it's called science also, right? To be open for fishing experiments. And I think we've seen over the last years some of these fishing experiments leading also to candidates that come up again and again. And this is something special about the last 5 to 10 years, that some candidates now are at the edge of being maybe clinically useful. I guess this is normal that people are doing all kinds of things, and be it academic sports or really convincing routes to finding a meaningful biomarker. I guess you cannot avoid it. Can I ask back, what's your key question? It sounded more like a comment.
The diversity in data we have around, I think it's very difficult to go retrospectively into those huge amounts of data.
No, I didn't really mean that.
Just a couple of years ago, when 23andMe was sending out wrong results by changing the input and output format by 180 degrees, this is not allowed to happen.
No, I didn't mean to reanalyze old data, but I meant that resources are sometimes quite scarce to analyze new data on specific biomarkers. So it's interesting to see how slow the process sometimes is, because people are very occupied with their routine daily activities or other activities. That's the only point I wanted to make. I do not want to mention or want to work up old data on all kinds of things. I just mean the high workload in society.
Can I comment on that before Florian comments on it? I think you make a very valid point. Paradoxically, pharmaceutical industry trials have one unknown in an equation, that is to show efficacy of a drug. And having a second unknown in the equation is somewhat a conundrum to be resolved. And then one solution to address that is that we need to have prospective trials where we validate the biomarker only in order to make it then applicable in whatever routine application or for clinical trials. And that is a very high hurdle for individual industry, but it needs to be raised as a gap in our concepts to achieve biomarker validation.
But imagine the placebo arms. Those samples could be really inform us about disease progression, how biomarkers are stable over time, and a lot of important things could be gained from them.
Can I comment on because I actually disagree a little bit by the constant focus on that proper prospective analysis are the only good way to study things. I do agree that it's of course always better to formulate guidelines for proper data collection, to do a standardized protocol for prospective analysis. I do agree with that that's better, but I think that the value of retrospective data is underestimated. If we can scale up, we could overcome some of the issues with retrospective data, even if it's just for hypothesis generating analysis that could actually learn you to do the prospective data collection better. To, for example, identify minimal requirements for the prospective data collections, to identify at least a set of variables that could be interested to focus on. Because in a prospective analysis, it's expensive. You cannot measure everything. And I believe that if you mine everything that is available today to the most optimal extent, your prospective data collection will be much, much better.
I had one comment to make, Florian van Rezong from Novartis, as a comment from your side and a question to the panel. How we want on one side collect data, big data, collective data from all data sources, and I agree, that's definitely the thing you do, and your MS Data Alliance is one of those examples. But how do we balance the scientific rigor on one side and the outcome of it? Otherwise we generate many anxiety, many aspirations that people believe, oh, this is true, and finally it's not the case, because I don't believe we have the method yet analyzing those large data yet. We don't have it yet. Simply that's a missing gap. But how do you see this moving on one side, large data, and this missing method and missing element here, because we're not there yet?
Yeah, so that's indeed a very challenging question, and we had already this discussion many times, but I think that there are some basic, simple things that could be done today to decrease the challenging efforts in the future. For example, we could teach clinicians that if they prescribe a drug, that instead of writing in an open letter the drug, sometimes with a typo, then an asterisk 2 milligrams, that they should put in one box a 2 and in another box mg. It's a basic thing. It's not rocket science, but it could increase a lot. It could also, I have taught PhD students of biomedical science that if they make an Excel file, that they should not leave the 2 first lines empty. Because a machine doesn't know that it's the 3rd line that they have to start. They start at the 1st line. And those basic things, by just making dictionaries of your data, these are the variables I'm collecting. And if I'm using M, this means male, then it's already a fast improvement of having data collection. That's what we mean with basic principle that make data fair, being findable. Open up your data, even if you just say, I have data, I have 10,000 patients and I'm collecting these variables. That's making your data findable. Reusable, it's having an informed consent that either makes reconsent possible or gives it quite broad, that you say, I am collaborating on these very general topics. So those are just basic things that could increase and that we can do today. So I've been in a conference on data leveraging where we addressed this in a multi-stakeholder setting, and we were all, again, we have to formulate research questions that are the focus of, and that's true, we have to do that. But at the same time, we can start with cataloging.
Don't you think the clinicians don't want to do that? Because it's all quite dependent on the electronic laboratory, the electronic patient records. So if it starts there,
exactly.
So if it's not possible to register in an easy way, so it's not about willing, it's also being able, and then to extract also those records from those highly protected systems. That's not an easy task.
No, it is not an easy task, but there are several initiatives arising that are doing that. For example,
yes, I know. Yeah.
And we could leverage those knowledge as soon as possible into the community of, for example, multiple sclerosis or any other community. So there is over 50 million being invested in an IMI project referred to as the European Health Data and Evidence Network, where they are mapping electronic health data on a common data model, which can be used for clinical research and which will in 4 years, 20 million people will be mapped on this common data model, and those data can be used. So it is indeed very challenging, and it's a multi-stakeholder effort.
So just from a standpoint of time, I think we've reached a point where we're going to need to take a break, but how many of you are presenting later today? Okay, awesome. So you'll have a chance to sit in on their presentations. The next, Julian, the timeline.
Yeah, so we have a 15-minute break now, and after that we divide into the two tracks. So people interested in neurology will stay here, and then we have a separate track on oncology and other topics that's upstairs, just half a flight upstairs.
So the neurology would stay here, oncology, and the other track is upstairs. Great, thank you very much. Really appreciate it.